Hello viewers, welcome to the course Elements of Genetics, and in today's part we shall be learning about multiple factor inheritance and multiple alleles. The quantitative traits are caused by Mendelian genes. What is important here in this particular part, that we are beginning a new chapter in the sense that the Mendelian genetics was about qualitative traits. That is, either it is tall or dwarf, either it is colored or colorless, either it is round or smooth. But here we shall get different shades of the traits which are quantitative traits. In 1909, Herman Nilsson Alleles from Sweden did a series of experiments with kernel color in wheat. Wheat is a hexaploid, the result of three different species producing a stable hybrid, an allopolyploid. There are thus three similar but slightly different genomes contained in the wheat genome called A, B and D. Let us recognize here the chromosome theory, that the organisms are made of certain set of chromosome or genome. The bread wheat, which is most commonly staple food of today, is basically a triploid which has three set of genomes from different parents. They are A, B and D. The quantitative traits are caused by Mendelian genes. Each genome has a single gene that affects kernel color, and each of these loci has a red allele and a white allele. We will call the red allele A, B and D, and the white alleles as small a, B and D. Inheritance of these alleles is partially dominant or additive. The amount of red pigment in the kernel is proportional to the number of red alleles present from 0 to 6. We shall recall that there are different colors of the wheat grain which determines its commercial value, and we can see that it is a golden yellow grain or it is a red grain, and the price of the wheat depends on that, and that grain color is basically a quantitative trait which was found by Nilsson Alleles and which forms the basis of the plant breeding today. This is a diagram which shows all the shades and the alleles which govern the kernel color or the grain color in wheat. Separating genetics from environment means that three experiments by Wilhelm Johansen from Denmark using the common bean, the Phaseolus vulgaris. Johansen coined the word genotype and phenotype, and the first Johansen experiment, he weighed a group of beans, then grew them up and weighed their progeny after selfing them. This is the chart which shows the quantitative variation, not the qualitative, but the quantitative variation in the grain weight of the Phaseolus vulgaris or the mung bean. The interpretation of this experiment is, in general, heavy parents means having heavy seed weight, gave heavy offspring, and the light parents gave light offspring. That is, there is a significant correlation between parent and offspring weights. However, there is also a considerable variation among the offspring weights. This is due to variation in both genetics and the environment. Here at this juncture, we are going to realize that the environment also plays a very important role in the expression of the gene. Whatever is genetically contained is called the genetic potential, and the expression will depend upon an interaction of genetic potential and the environment. The interpretation of these experiments that most offspring of extreme parents, that is very heavy or very light, are more average than their parents. This is a phenomena called regression to the mean. Extreme members of a population benefit from very lucky environmental condition which can't be inherited. The lucky environment condition means the favorable condition which allows genetic potentials to express fully. In Jonassen's second experiment, Jonasson worked on separating environment effect from genetic effect. He did this by inbreeding the beans for 10 generations, and inbreeding means doing the closest possible cross, in this case selfing them. The second experiment shows that half if the remaining heterozygosity disappears for each generation of selfing. These are the experiment of Jonasson through continuous inbreeding, and through continuous inbreeding he obtained a pure homozygosity. After 10 generation of selfing, the percentage of heterozygote is less than 1 out of 1000 of the original level, that is the extreme level of homozygosity. Results: Jonasson created 19 inbred lines. The inbred lines had some variation but less than the original random bred population. The remaining variation was due to the environmental variation. In fact, in all these experiments which show that the characters are quantitative in nature and they are governed by different alleles, and the pure homozygosity is being obtained through constant inbreeding. These Jonasson's third experiment showed that he started to work out the basis of artificial selection, how to improve a species as efficiently as possible. Then this procedure does not work for inbred populations. These are the Jonasson's pioneering and fundamental experiment which paved the way for future plant breeding. Selection is a procedure of creating a new genotype based on the visual characteristic of a plant or an organism through which they are selected, and then they are used for the crop improvement program through plant breeding methods. The additional genetic pattern, as we compare with the Mendel's pattern and other pattern, in case of Mendel there is a complete dominance, the 2 alleles per gene and one gene affects one trait. In other pattern there is an incomplete dominance, either there is a codominance, there are lethal alleles, there is a hierarchy of a dominance, there are multiple alleles, there are many traits which is pleiotropic, true or more genes affect one trait, that is gene interaction, and polygenic traits. By polygenic traits we mean that the one characteristic is governed by many genes, whereas in case of Mendel we always observed that the one gene controls expression of one phenotypic character. In another examples as compared to that of Mendel and other pattern, that in case of Mendel it could not observe gender specific trait. There were equal contribution from both parents. The trait expressed at same level and stage of life, and we could not visualize the environmental influence. In case of other pattern, there were sex influenced traits, sex limited traits, cytoplasmic inheritance was there, genetic maternal effect, genotypic imprinting was there were certain anticipations, and the role of environment was visible. The incomplete dominance is also an exception to the Mendel's law, and it is also concerned in case of multiple allele interaction. The incomplete dominance, neither allele masks the other, and both are observed as a blending in the heterozygous. Here we see that how in case of the Mendelian genetics, the cross between white and red always led to the red type, but the incomplete dominance shows that there are the production of pink type of genotypes because of the incomplete dominance. The multiple alleles, there are 3 or more alleles exist for one trait, and this is in case of the inheritance of the blood type, which we have type A, type B and type O, and the alleles governing are capital IA, capital IB and small I. These are the factors responsible for codominance. This is neither allele masks the other, so that the effects of both the alleles are observed in heterozygote without blending. Capital IA and capital IB are codominant. Capital IA and capital IB are completely dominant over small I. This is the diagrammatic representation of antigens on red blood cells. Inheritance of RH factor, that is rhesus factor, which is also very important in case of blood group. the phenotype, genotype, gene product and antibodies present. In case of Rh positive, it is capital R or small r. Rhesus protein is the gene product and antibodies are none. The Rh negative is both the small r, the gene product is none and antibodies are none unless exposed. This kind of genetic information is very useful in protecting the offspring or newborn baby from the lethal effects of the multiple alleles. The multiple alleles and codominance, the type A and Rh positive when crossed with type B and Rh negative, they result into the following kind of the combinations. The pleiotropic effects are called that when one gene affects many phenotypic characteristics. The alleles, the S type and S dash type, the gene product, the hemoglobin A and hemoglobin S. The cell shape is round, and in case of the another allele, it is sickled under low oxygen tension and in response to malaria susceptible and resistance in case of S and S dash genotype. This genetics is very useful in fighting with the malarial disease as well as the disease caused by sickle cell anemia under low oxygen pressure. The most profound example of the polygenic inheritance in case of skin color, and there are two genes which affect skin coloration, and this study has been conducted with African culture where we observe these quantitative variation in the skin color as observed in case of this table, that is white, light black, medium black, dark black and darkest black, 0 to 4 degree of variation. And this 0 to 4 degree of variation in case of genotype, it has been shown as all small a and small b, a combination of capital A and small b in case of light black, medium black, dark black and darkest black, and the pigmentation that is responsible for the skin coloration varies from 0 to 11, from 12 to 25, from 26 to 40, from 41 to 55 and 56 to 78% in case of the darkest black, and that is the pigmentation as governed and controlled by multiple alleles or the quantitative inheritance of the skin color. The polygenic inheritance is also shown here in case of this crossing, that a medium black woman marries to a darkest black man and the result of progeny can be darkest black, dark black or medium black as shown in the table, depending upon the various random combination of the multiple alleles. The interacting genes affecting a single characteristic, here it is commonly observed and have been studied in case of skin coloration in snake. The one gene orange is capital O, no orange is small o. The second gene B is black pigment, capital B and small b, and as a interacting genes affecting single characteristic of the variation and combination being shown here in the table will result into the different skin coloration of the snakes, which we can see in these photographs. The epistasis is another example of the multiple allele or quantitative inheritance, that an allele of one gene masks the expression of allele of another gene and expresses its own phenotype instead. Gene that masked is called epistatic gene, gene that is masked is hypostatic gene, and genes that code for enzyme that are upstream in a biochemical pathway usually exert epistasis, that is standing on. There are two kinds of epistasis, the dominant epistasis and recessive epistasis. Here the epistatic gene exerts its effect with homozygous recessive genotype, and they are expressed as W and M, that is enzyme 1 and enzyme 2, and they are ultimately responsible for the phenotypic expression of the flower color, that is pure white, pink or blue. The recessive epistasis is responsible for petal color in blue-eyed Mary plants, which we saw in the previous diagram, and the other colors which are possible are blue, magenta, white and white, depending upon the various combination of capital W, capital M, small m and their combinations as the multiple alleles. The duplicate recessive epistasis is also governed in petal color in hairbell flower, and the colors visible are blue, white, white and white as per the various combination of dominant and recessive alleles. The dominant epistasis again is the gene exerts its effect with the presence of a dominant allele, that is fruit color in summer squash. And here in case of summer squash, the yellow is capital Y, the recessive segregation is small y is green, and W is an inhibitor gene which inhibits either color. So whenever there is the W gene is present, it will inhibit the expression of color and the squash will be white, otherwise it would be the green or yellow depending upon the combination of the alleles. Dominant epistasis shows fruit color in summer squash as explained earlier, that is white, white, yellow and green. There is a interaction between sex and heredity, and this is a common knowledge that the gene of baldness is related with the X chromosome, which is responsible for the maleness in case of human beings, and the sex influenced character is determined by autosomal genes expression differs by the gender. This is the male pattern baldness, dominant in males and recessive in females. That is why the baldness is rampant and predominantly found in case of males only. The interaction between sex and heredity is another example in case of the cocks and the hens. There is one cock feathered male, the another is hen feathered female, and then third is hen feathered male. So here we see that sex limited characteristic determined by autosomal genes expressed only in one gender. Here this expression is case of male only and it is not seen in females. The interaction between sex and heredity is also governed and controlled by cytoplasmic inheritance. In another exceptions to the Mendelian genetics, the cytoplasmic inheritance has also been found to contribute, which is a characteristic only confined to the female parent, because the cytoplasm is contributed by the cytoplasm of the ovary. And in case of cytoplasmic inheritance, which is quite observed in the variegated leaves of the ornamental plant, the genes found on chromosomes of cytoplasmic organelles, and they are inherited from the maternal parent due to the contribution of cytoplasm in the ovum, and the leaf variegation caused by inheritance of variable chloroplast genotype. The interaction between sex and heredity has been explained in this diagram, that there are maternal genetic effects and the phenotype of offspring depend on genotype of the maternal parent, and the direction of the snail's shell coiling is determined by genotype of female parent. This is one of the most studied example in case of the cytoplasmic inheritance, and the direction of the coiling of the snail greatly depend upon the cytoplasmic inheritance. The example of certain genetic defects are also due to interaction between sex and heredity and the genomic imprinting. The expression of autosomal genes differs depending on whether they are inherited from the male or female parent. In case of a syndrome, that is Prader-Willi syndrome, deletion on chromosome 15 is inherited from father, and if it is inherited from a mother, it is known as Angelman syndrome, again which is a deletion on chromosome 15. The environmental effects are most profoundly observed in case of the rabbit skin color. The phenotype is dependent upon the presence of a specific environment. Here we see a color of a rabbit that is black and white, which is reared at 20 degrees Celsius or less, and the another rabbit which is pinkish white, which is reared at temperature above 30 degrees Celsius. The temperature sensitive product of the Himalayan allele is inactivated at high temperature. So these are the specific example of the gene environment interaction, that how the expression of a gene is controlled by the external environment, for example the temperature or the sunlight. Now the penetrance and expressivity, the characteristics like the smile of the face is also has been studied in genetics and known as penetrance and expressivity. The definition of penetrance is the percentage of individual with a given genotype who exhibit the phenotype, and the expressivity is extent to which genotype is expressed at the phenotypic level, may be due to allelic variation or environmental factors. Now in the adjoining diagram we see the various quantitative variation in the blue and white color and the sum of the coloration in between white and blue, which are variable penetrance, the variable expressivity and variable penetrance and expressivity, which will determine most finely manifested characteristics of the human face. Viewers, in today's part we actually studied about some of the variations from the Mendelian genetics, and the variations were specifically about the quantitative inheritance. We should recall that the Mendelian genetics had a qualitative genetics in the sense that the one gene controlled one character, and they differed qualitatively, that they were either tall or dwarf, they were either white or red, they were either smooth or round. So their characteristics were qualitatively separated. Here in today's part we shall study the characteristics which are quantitatively assorted. For example, skin color. We have all variation in the skin color, from extreme white to darkest black, and there are in between, that is the somewhat more white, somewhat less brown, somewhat dark black, or etc. These different shades of the skin color, when studied in African culture, we found all sort of variation, and in today's course program we saw that how multiple alleles governed these expression of the quantitative traits. Secondly, we observed the effect of the environment on the expression of the characteristics. The Nielsen-Alleles experiment clearly showed two things: the expression of the quantitative characters in the wheat kernel. The grain color of the wheat were the examples of the quantitative genetics, and also the effect of environment could also be visualized. It was most apparent in case of the coloration in case of rabbit, that the environmental temperature could also control the manifestation of the genetic trait. Finally, we observe that either it is the genes or the environment, as well as it is the interaction of the gene and environment. The gene is known as genetic potential, and final expression is controlled by genetic potential and interaction with the environment. We also observed that there are certain characteristics which are observed in case of epistasis, the pleiotropic effect, the inheritance of the blood group, the inheritance of the skin color, the inheritance of the skin color in snakes. All these examples led toward one understanding that either it is the polygenic control, multiple allele, and the quantitative inheritance. Thank you.